Have you ever felt that incredible surge of energy after a good meal? Like your body just switched into high gear?
Oh yeah, FM.
Or conversely, pushed yourself during a tough workout, hit that wall, and felt completely drained, muscles burning. We've all experienced these powerful shifts in our energy levels.
Absolutely, that fatigue, that burn, it's very familiar.
But what's truly orchestrating these changes inside us? What are the hidden gears turning, to create or deplete that vital energy?
That's the million-dollar question, isn't it? What's really going on under the hood?
Today, we're pulling back the curtain on the fundamental chemical processes that quite literally keep us alive, metabolism.
Mm-hmm.
We're talking about the constant building up and breaking down of molecules that powers every single cell.
It's happening constantly right now in all of us.
And this isn't just dry science, it's the intricate, beautifully designed engine of life packed with surprising facts and just unbelievable biological engineering. We're going to really unpack this.
That's exactly right. Our mission in this deep dive is to unravel this complex network of reactions. We'll start with a foundational overview of metabolism itself, then drill down into one of its most crucial pathways, glycolysis.
Glycolysis, okay.
You'll discover how your body takes glucose, the simple sugar from your food, and masterfully transforms it into the energy your cells desperately need.
Right, turning food into fuel.
Precisely. We'll explore how this entire process is meticulously regulated, both in the short term and over longer periods, and what happens when those finely tuned systems go awry, leading to various health conditions.
So not just what happens, but how it's controlled.
Exactly. Our goal isn't just for you to get a general idea, but to truly understand this information like an expert, grasping the interconnected pathways and the intricate signals that orchestrate life at its most fundamental level.
Like really mastering it.
Yeah, getting that deep professional level understanding.
And to guide us through this complex landscape, we've drawn from a comprehensive stack of foundational sources. These expert materials form the bedrock of our understanding today, ensuring we bring you the most accurate and insightful information directly from the heart of biochemical knowledge.
We've got the good stuff, the core texts.
So let's begin our journey by zooming out to get the big picture. When we think about individual chemical reactions in our bodies, it's easy to imagine them happening in isolation, like a single domino falling.
Right, like separate little events.
But here's where the brilliance of biology truly shines. In living cells, these reactions almost never occur on their own. Instead, they're meticulously organized into what our sources call multi-step sequences, what we know as metabolic pathways.
Pathways, exactly. Series of connected steps.
Take glycolysis, which we'll explore in detail soon. It's a perfect example. In these pathways, the product of one reaction instantly becomes the substrate, the raw material, for the very next reaction in the sequence.
One feeds directly into the next.
It's like a highly efficient biological assembly line, where each step seamlessly feeds into the next.
And it's not just a series of linear assembly lines. What's truly fascinating is that these individual pathways aren't isolated. They intersect and intertwine, forming what our sources describe as an integrated and purposeful network of chemical reactions.
A network. Okay, so it's all connected.
This dynamic, interconnected network is the very essence of metabolism. So metabolism, in its broadest sense, is the sum of all these chemical transformations happening within a cell, a tissue, or even your entire body. It's a constant dynamic state of flux, always adapting.
It's always changing, always working.
Beyond linear pathways, metabolism also beautifully incorporates cycles. These are pathways where a specific component is regenerated at the end, allowing the entire cycle to continue repeatedly.
Like the Krebs cycle, maybe.
Exactly like the Krebs cycle or the TCA cycle. That's a perfect example. This demonstrates the incredible efficiency and elegant interconnectedness of biological systems.
Sufficient design.
To truly grasp the vastness and complexity of this system, our sources recommend visualizing it using a metabolic map. Think of it like a sprawling, intricate subway system, but for molecules.
A map for molecules. I like that.
This map, similar to Figure 8.2 in our source material, is invaluable. It helps us trace the connections between different pathways, observe the deliberate movement of metabolic intermediates, what we call metabolites.
Metabolites, okay.
And, crucially, understand the ripple effect when a pathway is blocked. This blockage could be due to something external, like a drug, or something internal, like an inherited enzyme deficiency.
So you can see the knock-on effects.
Precisely. Every dot on this map represents a specific chemical, and every line is an enzymatic reaction, showing how everything is interwoven.
And when we talk about all the metabolites in an organism, that complete set of chemical compounds, it has its own specific name, the metabolome.
A metabolite,
Yeah.
it's a snapshot.
It's a powerful concept because it offers a snapshot of the precise chemical state of an organism at any given moment, reflecting both its current activity and its health.
Like a chemical fingerprint.
So, what's the big takeaway from understanding this metabolic map? It's the ultimate cause and effect diagram for your body. Imagine a traffic jam on one street. This map instantly shows you how it bottlenecks other parts of the city.
That's a great analogy.
That's how a single enzyme deficiency, or even a medication, can ripple through your entire system, revealing a level of interconnectedness far beyond what we typically imagine when we think of body chemistry.
It's way more connected than you might think.
It's the blueprint for how you function and why subtle changes can have profound effects.
Absolutely fundamental.
Now that we have that grand overview, let's break metabolism down into its two primary yet opposing categories: catabolism and anabolism.
The two sides of the coin.
Think of them as the two sides of a perfectly balanced coin, constantly working in opposition and in concert to keep you going.
Okay, so catabolism. In essence, this is the breaking down side. These are the degradative processes.
Degradative, like demolition.
Kind of, yeah. Catabolic pathways take complex molecules like the proteins in your steak, the carbohydrates in your pasta, or the lipids in your avocado, and systematically break them down into much simpler ones, such as carbon dioxide, ammonia, and water.
Okay, breaking big stuff into small stuff.
Exactly. The ultimate purpose of catabolism is to capture the chemical energy, primarily in the form of ATP, from the degradation of these energy-rich fuel molecules.
ATP, that's the key energy currency, right?
That's the one. This ATP generation happens in three distinct yet interconnected stages, beautifully illustrated in figure 8.3 of our source. The first stage is the hydrolysis of complex molecules.
Hydrolysis, breaking with water.
Right. This is where the large, intricate molecules you consume are initially broken down into their fundamental building blocks. For instance, proteins are chopped into individual amino acids.
Okay.
Complex polysaccharides, like starch, are broken into simple monosaccharides, like glucose. And fats, or triacylglycerols, are broken down into free fatty acids and glycerol.
So you start with the big, complex pieces and reduce them to their fundamental components. Then what's stage two?
Stage two is the conversion to simple intermediates. These smaller building blocks are then further degraded. They're primarily converted into a simpler common intermediate called acetyl coenzyme A, or acetyl-CoA.
Acetyl-CoA. I've heard of that one.
It's central. At this stage, a small but significant amount of ATP is actually captured, but it's just a fraction compared to the vast energy harvest that's coming next.
Just a little bit of energy here.
Precisely. The real energy windfall comes in the third stage, the oxidation of acetyl coenzyme A. This is where the tricarboxylic acid cycle, or TCA cycle, also widely known as the Krebs cycle, takes center stage.
Ah, the famous Krebs cycle.
It's truly the final common pathway for the complete oxidation of virtually all fuel molecules that produce acetyl-CoA. During this stage, massive amounts of ATP are generated through a process called oxidative phosphorylation.
Oxidative phosphorylations. That sounds important.
It's huge. This happens as electrons, carried by highly energetic molecules like NADH and FADH2, flow through a series of specialized protein complexes within your mitochondria, ultimately to oxygen.
In the mitochondria, the powerhouses.
Exactly. It's an incredibly efficient energy-generating factory within your cells, essentially where the bulk of your body's energy is produced under aerobic conditions, meaning with oxygen.
Okay, so summing up catabolism, it's breaking down, it's oxidative. What else?
Right. These pathways are typically oxidative, meaning they involve the removal of electrons from molecules. This often requires oxidized coenzymes like NAD+ to accept those electrons.
NAD+ acts like an electron shuttle.
It does, and a key characteristic of catabolism is that it's a convergent process. Think of it like deconstructing a wide variety of complex machines, a car, a washing machine, a computer, to extract energy from their components.
Okay.
No matter how different they started, you break them all down into a few common, simple end products, like raw materials or basic parts.
Right, you end up with similar basic bits.
Similarly, your body takes a vast array of complex fuel molecules and transforms them into a few common metabolic end products, primarily acetyl-CoA, to efficiently extract energy. It funnels everything down.
Now let's flip the coin and talk about anabolism. If catabolism is the body's demolition crew, constantly tearing things down for parts.
Then anabolism is the construction crew.
Exactly. Anabolism is all about building up. These are the synthetic processes where your body constructs complex products, like the glycogen you store in your liver and muscles, the proteins that make up your tissues, or the various lipids in your cell membranes.
All the complex structures.
From simple precursors, such as glucose or individual amino acids.
In direct contrast to catabolism, which releases energy, anabolic reactions are inherently endergonic, meaning they require an input of energy to proceed.
So they cost energy, they don't make it.
Correct. This necessary energy is typically supplied by the hydrolysis of ATP to ADP and inorganic phosphate, pi. So catabolism generates the ATP, and anabolism consumes it.
It's a beautiful, symbiotic metabolic partnership, a true give and take.
It really is. Furthermore, anabolic reactions often involve chemical reductions, where electrons are added to molecules to build them up.
The opposite of oxidation and catabolism.
Precisely. The reducing power for these reactions, supplying those electrons, is most frequently provided by another key electron donor called NADPH.
NADPH related to NADH.
Yep, it's essentially a phosphorylated version of NADH, specifically adapted for biosynthetic reactions for building things.
Okay, specialized for construction.
And unlike catabolism, which is a convergent process, anabolism is a divergent process. As figure 8.4 in our source illustrates, you start with a relatively small number of simple biosynthetic precursors, perhaps just glucose or amino acids.
A few basic building blocks.
And from those, your body can create an astonishingly wide variety of complex products. This includes all the different proteins, lipids, carbohydrates, and nucleic acids that form the intricate architecture and machinery of your cells and tissues.
Wow, from simple to complex.
It's incredible diversity from simple starting points.
And this constant dynamic equilibrium between catabolism and anabolism is why this entire metabolic dance matters so much to you, right?
Absolutely, it's fundamental.
It's what allows your body to both generate the immediate energy it needs to function and simultaneously build, repair, and maintain everything from your muscles and organs to your hormones and enzymes.
It's the balance of life.
It's a continuous, perfectly orchestrated construction and deconstruction project happening inside you every second of every day, ensuring your body is always in a state of dynamic balance.
A constant balancing act.
Okay, given the incredible complexity of metabolism, with countless pathways constantly at work, it should come as no surprise that these processes need to be coordinated with astonishing precision.
Yeah, it can't just be chaos. There has to be control.
Your cells aren't just randomly breaking things down or building things up. They're constantly responding to the body's moment-to-moment energy and synthesis needs. How do they know what to do?
That's where regulation comes in, the master coordinators.
Let's talk about those.
All right, so the first level of control happens right inside the cell, with what we call intracellular signals. These are the rapid, immediate adjustments.
Quick responses.
Yeah, really quick. These signals are how a metabolic pathway can instantly adjust to its immediate environment. For example, the rate of a reaction might be influenced by the simple availability of its substrates.
Makes sense. No fuel, no go.
Right. Or it could be affected by product inhibition, where the end product of a pathway actually signals back to an earlier enzyme, telling it to slow down because enough has been made.
Like feedback control.
Exactly, negative feedback. Then there are allosteric activators or inhibitors, clever molecules that bind to an enzyme at a site other than its active site, subtly changing its shape and either boosting or slowing its activity.
So they're like dimmer switches for enzymes.
That's a good way to put it.
Yeah.
These intracellular signals typically elicit very rapid responses, making them crucial for the moment-to-moment fine-tuning of metabolism, much like a car's engine immediately adjusts fuel flow based on how hard you're pressing the pedal.
Instant adjustments based on immediate needs. But individual cells aren't isolated entities operating in a vacuum, are they?
Not at all. They're part of a community.
They function as part of a vast, interconnected community of interacting tissues, organs, and systems. So beyond those internal, immediate adjustments, a sophisticated communication system has evolved to coordinate the functions of the entire body.
Long-range communication.
Think of it as the ultimate biological network, a complex conversation constantly happening throughout your system.
Absolutely. This ability to respond to intercellular signals is essential for the development and survival of complex organisms like us. These signals provide long-range integration of metabolism, allowing different organs to work in harmony.
Getting everyone on the same page.
Right. They often lead to responses that are a bit slower than intracellular signals, sometimes even involving changes in gene expression that can take hours or days to manifest.
Slower, but maybe more profound changes.
Often, yes. The regulatory signals that inform an individual cell about the metabolic state of the entire body include familiar messengers like hormones, neurotransmitters, and even the simple availability of nutrients circulating in the bloodstream.
Like insulin telling cells about blood sugar.
Perfect example. While direct cell-to-cell contact and specialized junctions allow for some communication between adjacent cells for systemic energy metabolism, the most important route of communication is chemical signaling between cells.
Chemical messages traveling around.
Primarily occurs via blood-borne hormones or neurotransmitters. These are the signals that tell your brain when your muscles need to get ready for action, or how your liver knows when to store energy versus when to release it into the bloodstream for other tissues to use.
So coordinating the whole body's energy strategy.
It's the central command system for your entire metabolic state, ensuring all systems are aligned.
So we have these crucial extracellular messengers like hormones and neurotransmitters acting as the initial signals. Your cell's receptors are the sophisticated signal detectors.
Listening posts on the cell surface.
When one of these external signals, or a ligand, binds to its specific receptor on the cell surface, it's like pressing a button that initiates a whole cascade of reactions inside the cell.
Triggers a response chain.
But how does that external signal actually translate into an internal cellular response, especially when the signal itself doesn't enter the cell?
Good question.
Okay.
The message needs to get inside somehow.
This is where second messenger molecules come into play. They're called second messengers because they bridge the gap between the original extracellular messenger, the hormone or neurotransmitter, and the ultimate intracellular effect.
They relay the signal internally.
They effectively convert the external signal into a specific cellular response.
And among the most widely recognized second messenger systems, one is particularly important in regulating intermediary metabolism, the adenyl cyclase system.
Adenyl cyclase.
Okay. This system often involves the binding of ligands like epinephrine, adrenaline, or glucagon to specific receptors on the cell membrane known as G protein-coupled receptors, or GPCRs.
GPCRs. Heard of those too. They're involved in a lot.
They are incredibly common. These GPCRs are designed like molecular antennas with the part that binds the external signal, a segment that spans the cell membrane 7 times, actually, and an internal part that interacts with what we call trimeric G proteins.
G proteins? The link.
The link, exactly. It's worth noting that not all metabolic regulators use GPCRs. For example, insulin, another key regulator of metabolism, binds a different type of receptor, a membrane tyrosine kinase receptor, which triggers a different, more direct signaling pathway.
So multiple ways to get the message across.
Definitely. But back to the adenyl cyclase system. When a chemical signal binds to a GPCR, such as those for adrenaline or glucagon, it triggers either an increase or a decrease in the activity of adenyl cyclase, or AC.
Okay, turns AC up or down.
Right. This AC enzyme is embedded in the cell membrane, and its job is to convert ATP into 3-in-wall adenosine monophosphate, more commonly known as cyclic AMP, or CAMP.
Yeah, AMP, that's the second messenger here.
That's the key second messenger in this system. Each hormone or neurotransmitter typically binds to a unique type of GPCR, which means cells responding to multiple signals will have several different GPCRs, each capable of linking to AC.
So how does the GPCR actually tell adenyl cyclase what to do? This is where our guanosine triphosphate-dependent regulatory proteins, or G proteins, come in.
The G proteins are the crucial middlemen.
The effect of an activated GPCR on second messenger formation isn't direct. It's mediated by these specialized trimeric G proteins, which literally link the receptor to adenyl cyclase.
They bridge that gap.
Think of it like this. In its inactive form, a part of the G protein, the alpha subunit, is bound to GDP. But when a ligand binds to the GPCR, it causes a change.
A conformational change, yeah.
That triggers the replacement of that GDP with a GTP molecule on the G protein's alpha subunit. This GTP-bound alpha subunit then physically dissociates and moves to adenyl cyclase, directly influencing its activity.
Activates or inhibits AC.
What's really powerful here is the amplification effect. A single activated receptor can activate many G protein alpha subunits,
which in turn can activate or inhibit many adenyl cyclase enzymes,
generating a large number of CAMP molecules. This means a tiny external signal can create a huge internal response.
Massive signal amplification. It's very efficient.
And this adds another layer of precise control, doesn't it?
It does. The ability of a hormone or neurotransmitter to either stimulate or inhibit adenylate cyclase depends on the specific type of G protein alpha subunit linked to the receptor.
Ah, different types of G proteins.
Exactly. For instance, some G alpha proteins called G stimulate AC, while others called G inhibit the enzyme. This dual control allows for very fine-tuned regulation.
Stimulatory and inhibitory signals.
Crucially, the actions of the active G alpha protein are inherently short-lived. The G alpha subunit itself has an intrinsic timer, a built-in enzyme activity called GT-pace activity.
A self-destruct timer.
Sort of. It rapidly hydrolyzes its bound GTP back to GDP. This quickly inactivates the G alpha subunit, causing it to detach from AC and reassociate with its other subunits, effectively turning off the signal.
So the signal doesn't just stay on forever.
Right. This rapid inactivation ensures that the cellular response is transient and tightly controlled, only lasting as long as the extracellular signal is present.
And this system is so critical that we can even see its disruption in the context of devastating diseases, offering a fascinating, if grim, real-world connection.
Yeah, the cholera and pertussis examples are striking.
Consider the toxins from Vibrio cholerae, which causes cholera, or Bordetella pertussis, which causes whooping cough. These toxins cause inappropriate activation of adenyl cyclase by essentially messing with different G proteins.
They hijack the system.
In cholera, the toxin prevents the off switch, that GT-pace activity on the stimulating G's alpha protein in intestinal cells, from working.
So G stays permanently on.
Right. Since GLIS is meant to stimulate AC and then quickly turn itself off, preventing its deactivation means AC remains continuously active, leading to nonstop CMP production and the severe diarrhea characteristic of cholera.
A stuck-on switch, causing massive fluid loss.
With whooping cough, the pertussis toxin inactivates the inhibitory G alpha protein in respiratory tract cells.
Takes out the brakes.
Exactly. Since G normally inhibits AC, inactivating it also leads to inappropriate and continuous activation of AC because the normal off signal is gone. This contributes to the severe symptoms of whooping cough.
So in both cases, you get way too much CMP, just through different mechanisms.
It illustrates just how profound the physiological impacts of specific molecular interactions can be, affecting everything from widespread disease symptoms to the precise effectiveness of medications targeting these pathways.
Tiny molecular changes, huge consequences. So once KMP is produced as a second messenger, what happens next inside the cell? How does it actually cause a response?
Right. CMP itself isn't the final actor.
The critical step in the CAMP second messenger system is the activation of a family of enzymes called CAMP-dependent protein kinases, most notably protein kinase A, or PKA.
PKA, another key player.
As figure 8.8 shows, CAMP activates PKA by binding to its regulatory subunits, which then causes the release of its catalytically active subunits.
So CAMP unlocks PKA's active part.
Exactly. These now active subunits get to work, transferring phosphate groups from ATP to specific serine or threonine residues on various target proteins within the cell.
Phosphorylation again, adding phosphate groups.
Yes, and this phosphorylation is like flipping a molecular switch. It can either activate or inhibit those target proteins, depending on the protein, leading to a specific cellular response dictated by the original hormone signal.
Okay, so PKA does the actual work of changing protein activity.
But just as important as putting those phosphate groups on is taking them off. This is where protein phosphatases come in.
The cleanup crew.
Essentially, yes. They act as the crucial off switch by removing the phosphate groups that protein kinases like PKA just added. They hydrolytically cleave those phosphate bonds,
Breaking them off.
ensuring that any changes in protein activity induced by phosphorylation are not permanent. This provides a critical on-off mechanism, allowing the cell to reset and respond to new signals.
Like a dimmer switch, kinases turn it up, phosphatases turn it down.
That's a perfect analogy. It ensures dynamic responsiveness.
And what about the CAMP itself? Does it hang around?
No, it's dealt with quickly too.
Yeah.
Adding another layer of essential control, CAMP itself is rapidly terminated by an enzyme called CAMP phosphodiesterase.
Phosphodiesterase, PDE.
Right. This enzyme quickly cleaves the cyclic bond of CAMP, converting it to 5-AMP. Critically, 5-AMP is not an intracellular signaling molecule.
So it kills the signal.
Exactly. Therefore, the effects of any hormone or neurotransmitter-mediated increases of CAMP are quickly terminated once the extracellular signal is removed. This rapid hydrolysis ensures a swift, precise, and transient cellular response.
Tight control over the signal duration.
And here's a fascinating connection to your morning routine. CMP phosphodiesterase is actually inhibited by caffeine, a methylxanthine compound found in coffee and tea.
Caffeine inhibits the enzyme that breaks down CAMP.
That's right. So when you drink coffee, part of the reason you feel that stimulating effect is because caffeine is prolonging the action of CAMP, essentially keeping those cellular signals, like those triggered by adrenaline perhaps, active for longer.
It's like caffeine is holding the on switch active for a bit longer by blocking the off enzyme.
Exactly. It's like the phosphodiesterase is trying to reset the dimmer switch,
Right.
but caffeine is jamming it in the on position, extending the cellular wake-up call.
Fascinating how everyday things interact with these pathways. All right, now that we've laid the foundational understanding of metabolism and its sophisticated regulation, let's finally dive into the star of our show today.
Glycolysis.
The main event.
This pathway is a cornerstone of cellular energy production, utilized by virtually all tissues in your body for the oxidation of glucose.
It's pretty much universal.
It provides both immediate energy in the form of ATP and essential intermediate molecules for other metabolic pathways. It truly sits at the hub of carbohydrate metabolism because nearly all sugars, whether they come directly from your diet or from other catabolic reactions in your body, can ultimately be converted into glucose to feed into this central pathway.
All roads lead to glycolysis in a way.
Glycolysis itself can proceed under two main conditions. Aerobically, meaning in the presence of oxygen, or anaerobically, without sufficient oxygen.
With or without O2.
In cells with mitochondria and an adequate supply of oxygen, pyruvate is the final product of what we call aerobic glycolysis. This 10-reaction series is termed aerobic because oxygen is ultimately needed to reoxidize the NADH that's formed during one of the key steps.
NADH needs to be recycled, and oxygen helps with that aerobically.
Aerobic glycolysis then perfectly sets the stage for pyruvate to be further processed into acetyl-CoA, which, as we mentioned earlier, is a major fuel for the powerful TCA cycle.
Leads into the big energy production phase.
Alternatively, if there's no oxygen or not enough, pyruvate is quickly reduced to lactate, as NADH is simultaneously oxidized back to NAD plus CYP.
That recycles the NADH anaerobically.
This conversion of glucose to lactate is called anaerobic glycolysis, and it's absolutely vital because it allows your cells to produce immediate ATP in tissues that either naturally lack mitochondria, like your red blood cells, or parts of your eye,
Tissues that can't use oxygen efficiently or at all.
or in cells that are temporarily deprived of sufficient oxygen, a critical condition known as hypoxia.
Like during intense exercise.
Oh.
But before glucose can embark on its glycolytic journey, it must first gain entry into the cell. How does it get across that membrane?
Right, it can't just wander in.
Glucose cannot simply diffuse directly through the cell membrane because it's a polar molecule. It requires specific transport systems. There are two primary ways glucose enters cells: a sodium, Na plus Se, an ATP-independent transport system, and Na plus Le, an ATP-dependent cotransport system.
Two main doors into the cell for glucose.
Let's start with the sodium and ATP-independent transport system, which is a form of facilitated diffusion, a passive process.
Facilitated diffusion means it needs help but no direct energy cost.
Exactly. This is mediated by a family of 14 distinct glucose transporter, or GLUT, isoforms, ranging from GLUT1 to GLUT14, found embedded in cell membranes.
GLUT transporters. Lots of them.
These are monomeric protein transporters that exist in two conformational states. When extracellular glucose binds to the transporter, it causes the protein to change its shape, effectively shuttling the glucose across the cell membrane down its concentration gradient.
From high concentration outside to lower inside.
Right. Crucially, this process requires no direct energy expenditure by the cell itself. Because these GLUT transporters move one molecule at a time, they are classified as uniporters.
Just moving glucose one way down the slope.
What's truly fascinating about these GLUT transporters is their tissue-specific expression, meaning different tissues have different types of GLUTs, each with specialized functions tailored to that tissue's needs.
Different tissues, different doors. Makes sense.
For instance, GLUT3 is the primary isoform found in neurons, which makes perfect sense, given the brain's continuous and high demand for glucose.
Brain needs glucose constantly.
GLUT1 is abundant in red blood cells and at the blood-brain barrier, ensuring glucose delivery to these vital areas, but it's less common in adult muscle. Now, here's a critical link to a major metabolic hormone, GLUT4.
Ah, the insulin one.
That's the one. GLUT4 is highly abundant in muscle and adipose fat tissue, and its presence on the cell surface and overall activity are significantly increased by insulin.
So insulin basically opens the GLUT4 doors in muscle and fat.
Pretty much, yeah. It causes more GLUT4 transporters to move to the cell surface. This is a key mechanism for how insulin helps regulate blood sugar by promoting glucose uptake into these major energy-consuming and storing tissues after a meal.
Crucial for blood sugar control.
Then there's GLUT2, which is abundant in the liver, kidneys, and pancreatic beta cells. Uniquely, GLUT2 can transport glucose into these cells when blood glucose levels are high, but also out of them when levels are low, like during fasting.
A two-way street for glucose in the liver?
Helping the liver maintain overall blood glucose homeostasis for the rest of the body. And finally, GLUT5 is a bit of an outlier. It's primarily a transporter for fructose, not glucose, found mainly in the small intestine where it absorbs dietary fructose and testes.
So, mainly for fructose. Okay. What's the other transport system?
In contrast to the passive GLUT system, we have the sodium and ATP-dependent cotransport system. So this is an energy-requiring process that actively transports glucose against its concentration gradient.
Uphill, from low concentration outside to high inside.
Exactly. It achieves this by coupling glucose transport with the simultaneous movement of sodium ions down their electrochemical gradient.
So glucose hitches a ride with sodium flowing downhill.
Right. This type of coupled transport, where two molecules move in the same direction, is called symport. The crucial detail here is that the sodium gradient itself, which powers this cotransport, is maintained by the NAB plus TXA plus ATPase.
The sodium-potassium pump?
An enzyme that actively pumps sodium out of the cell using ATP. So this makes glucose transport an indirect or secondary active transport system. It uses energy, but indirectly.
Okay. The pump sets up the gradient, and the cotransporter uses the gradient.
Precisely. This type of glucose transport is found in the epithelial cells of your intestine, allowing you to efficiently absorb glucose from your diet even when concentrations are low. In the renal tubules of your kidneys, where glucose is reabsorbed back into the blood, preventing its loss in urine, and in the choroid plexus, part of the blood-brain barrier.
So why is understanding how glucose gets into your cells so crucial? Seems like a basic step.
It is basic, but it's fundamental. This initial gateway directly dictates whether your body can effectively use glucose for immediate energy, store it for later, or if it contributes to persistently high blood sugar levels.
It controls the supply.
Exactly. This foundational understanding is absolutely critical to grasping conditions like diabetes and the broader aspects of nutrition. It's the very first, highly regulated step that glucose must pass through on its journey to become usable energy for your cells.
Okay, glucose is inside. Now, glycolysis truly begins. You mentioned two stages, an investment phase first.
Right. This 10-step pathway can be divided into two main stages.
Yeah.
An initial energy investment phase, which includes the first 5 reactions, and then an energy generation phase.
Gotta spend energy to make energy.
You got it. In this investment phase, your cell actually uses ATP to synthesize specific phosphorylated intermediates, effectively priming the glucose molecule for later, larger energy extraction.
Okay, step one of the investment.
The very first step, the phosphorylation of glucose, is absolutely critical. Glucose is irreversibly phosphorylated to glucose 6-phosphate.
Adding a phosphate group, why is that so critical?
Because phosphorylated sugar molecules, like glucose 6-phosphate, are too polar to easily diffuse back out of the cell, and they lack specific transmembrane carriers that allow them to exit.
So adding the phosphate traps it inside.
Exactly. It elegantly traps the sugar as cytosolic glucose 6-phosphate, committing it to further metabolism within the cell. Mammals have 4 different isozymes of the enzyme hexokinase I through 4V that catalyze this initial phosphorylation.
4 versions of the same enzyme, hexokinases.
Let's first look at hexokinases I3. These are found in most tissues and are one of 3 key regulatory enzymes in glycolysis. They're primarily inhibited by their product, glucose 6-phosphate.
Feedback inhibition again?
Right. It accumulates when the cell's immediate metabolic needs for glucose decrease. A crucial characteristic of hexokinases is their low Michaelis constant.
Low commands means high affinity. They grab glucose easily.
Very high affinity. This allows them to efficiently phosphorylate glucose, even when its concentrations in the tissue are quite low, as seen in figure 8.13. However, they also have a relatively low maximal velocity, Vmax.
Low Vmax means they work steadily but not super fast.
Correct. This is important because it prevents these enzymes from trapping excessive amounts of cellular phosphate in the form of phosphorylated glucose, or phosphorylating more glucose than the cell can immediately use. They ensure a steady, controlled uptake. They also have a broad specificity, meaning they can phosphorylate other hexases like fructose in addition to glucose.
Okay, so hexokinases I3 are the general workhorses in most tissues. What about hexokinase the fourth? You mentioned the liver handles things differently.
Exactly. That's where glucokinase or hexokinase the fourth comes in. This isozyme is predominant in liver parenchymal cells and, crucially, in pancreatic beta cells.
Liver and pancreas beta cells, why there?
In those beta cells, glucokinase functions as a vital glucose sensor, directly determining the precise threshold at which insulin is secreted in response to blood glucose levels.
It tells the pancreas when to release insulin.
Wow. It also acts as a glucose sensor in hypothalamic neurons, playing a key role in the body's adrenergic response to hypoglycemia, or low blood sugar. In the liver, glucokinase's primary role is to facilitate rapid glucose phosphorylation, especially during times of high blood glucose, like after a carbohydrate-rich meal.
So it helps the liver soak up glucose when there's a lot of it.
Precisely. Glucokinase differs significantly from the other hexokinases in its kinetics, which speaks volumes about its role. It has a much higher chemomemer.
Higher creatinine, lower affinity. It only works well when glucose is abundant.
You got it.
Yeah.
This ensures that glucokinase primarily becomes active and phosphorylates glucose only when the intracellular glucose concentration in liver cells is elevated, such as during the brief period right after a carbohydrate-rich meal when high levels of glucose are delivered to the liver via the portal vein.
Smart design. And what about its speed, its Vmax?
And here's the clever part. Glucokinase also has a high Vmax. This allows the liver to effectively remove the flood of glucose delivered by the portal blood, preventing large amounts of glucose from entering the systemic circulation and thereby minimizing post-meal hyperglycemia.
So it can handle a large influx quickly.
And remember that GLUT2 transporter we just talked about, the two-way one in the liver. It ensures that blood glucose equilibrates rapidly across the hepatocyte membrane, perfectly facilitating this high-capacity uptake by glucokinase.
The transporter and the enzyme work together perfectly.
The regulation of glucokinase is also unique and sophisticated. Unlike the other hexokinases, it's not directly inhibited by its product, glucose 6-phosphate.
No feedback inhibition. How is it controlled then?
Instead, it's indirectly inhibited by fructose 6-phosphate, which is in equilibrium with glucose 6-phosphate, and indirectly stimulated by glucose itself. This precise regulation is achieved by reversible binding to a hepatic protein called glucokinase regulatory protein, or GKRP.
GKRP, a regulator protein.
In the presence of fructose 6-phosphate, glucokinase binds tightly to GKRP and gets translocated into the cell's nucleus, effectively inactivating the enzyme, as shown in figure 8.14.
Hides it in the nucleus when there's enough product downstream.
Basically, yes. But when glucose levels in the blood, and consequently in the hepatocyte, increase, glucokinase is released from GKRP and re-enters the cytosol, ready to phosphorylate glucose. It's a precise molecular switch that elegantly responds to your body's overall glucose levels.
That's incredibly elegant control.
What's particularly important here clinically is that inactivating mutations of glucokinase are the cause of a rare but significant form of diabetes called maturity-onset diabetes of the young type 2, or MODY2.
MODY2, caused by faulty glucokinase.
Yes. This condition is characterized by impaired insulin secretion, because the beta cell sensor isn't working right, and chronic hyperglycemia, underscoring just how crucial this single enzyme is to the body's overall blood sugar control.
Wow. So this initial step and the fascinating functional differences between hexokinase and glucokinase reveals a deep insight. It dictates how your various tissues prioritize and handle glucose.
It sets the stage for everything else.
Some tissues, like your brain, need a constant high-affinity supply via hexokinase I3, while others, like your liver using glucokinase, are designed to buffer large influxes after a meal. This foundational step plays a direct and critical role in your blood sugar control, and as we just heard, is directly implicated in the development of certain types of diabetes.
It's the very first strategic decision your cells make about incoming glucose.
Okay, what's next after trapping the glucose as glucose 6-phosphate?
Following that critical initial phosphorylation, glucose 6-phosphate then undergoes a straightforward isomerization to fructose 6-phosphate.
Isomerization, just
Right.
rearranging the atoms.
Exactly. Catalyzed by the enzyme phosphoglucose isomerase. This reaction, depicted in figure 8.15, is readily reversible and is not a rate-limiting or regulated step in the pathway. It's a quick, simple rearrangement, essentially preparing the molecule for the next key step.
Getting it ready for the big commitment step, maybe? Hashtag fructose 6-phosphate phosphorylation.
You anticipated it perfectly. Now we arrive at what is arguably the most critical regulatory step in all of glycolysis, the phosphorylation of fructose 6-phosphate.
Another phosphorylation, so another ATP used.
Yes, this is the second ATP investment. This irreversible reaction is catalyzed by phosphofructokinase 1, or PFK1.
PFK1, this sounds like a really important one.
It absolutely is. Our sources emphasize that PFK1 is the most important control point, the rate-limiting and the committed step of glycolysis.
Rate-limiting and committed, meaning once this happens, there's no going back.
Pretty much. Once this step happens, the glucose molecule is fully committed to being broken down through glycolysis, as you can see in figure 8.16. PFK1's activity is meticulously controlled by the concentrations of its substrates, ATP and fructose 6-phosphate, as well as by other very important allosteric regulatory molecules.
Okay, how is it controlled? You mentioned ATP.
PFK1's regulation by intracellular energy levels is remarkably intuitive. It's allosterically inhibited by elevated levels of ATP.
Makes sense. High energy, slow down glycolysis.
Exactly. High ATP is a clear signal that the cell has abundant energy and therefore doesn't need to break down more glucose. Similarly, high levels of citrate, an intermediate of the TCA cycle, also inhibit PFK1.
Citrate from the Krebs cycle. Why does that inhibit PFK1?
Because high citrate tells the cell that plenty of fuel is already being processed through the later energy-generating cycles. So this inhibition actually encourages the diversion of glucose towards glycogen synthesis for storage rather than immediate breakdown. It's like saying, we have plenty of fuel downstream, maybe store this glucose instead.
Smart coordination. What activates PFK1 then?
Conversely, PFK1 is powerfully activated allosterically by high concentrations of AMP.
AMP, low energy signal, right?
Exactly. AMP signals that the cell's energy stores are depleted because ATP has been broken down to ADP and then AMP, and it desperately needs to generate more ATP. So AMP yells go to PFK1.
Okay, ATP and citrate inhibit, AMP activates.
Right.
What else? You mentioned another regulator.
Yes, the most potent activator, PFK1, the ultimate master switch, is a molecule called fructose 2-kinase-6-bisphosphate.
Fructose 2-6-bisphosphate, not the 1-sugar-6 we just made.
Correct. It's a different molecule,
Yeah.
specifically made as a regulator. It's so powerful that it can activate PFK1 even when ATP levels are high, overriding the stop signal from ATP.
Wow, it overrides ATP inhibition. How is it made?
Now, fructose 2-6-phosphate isn't an intermediate of glycolysis itself. It's specifically formed from fructose 6-phosphate by another enzyme, phosphofructokinase 2, or PFK2.
PFK2. So there are two PFKs.
Yes, but PFK2 is very different. And here's where it gets truly ingenious. Unlike PFK1, PFK2 is a unique bifunctional protein.
Bifunctional. Two functions in one protein.
That means it has two distinct enzymatic activities on a single polypeptide chain. It has kinase activity, which produces fructose 2-6-bisphosphate from fructose 6-phosphate, and phosphatase activity, which dephosphorylates it back to fructose 6-phosphate.
Whoa, it makes the activator and breaks it down, all in one enzyme.
Exactly. It's a built-in control mechanism for the level of this potent activator.
How is PFK2 itself regulated then?
The regulation of this bifunctional PFK2 protein is quite sophisticated and elegantly tissue-specific. In the liver lysozyme, for example, phosphorylation of PFK2 actually inactivates its kinase domain while simultaneously activating its phosphatase domain, as depicted in figure 8.7a.
So phosphorylation in the liver decreases the activator, fructose 2-6-bisphosphate.
Precisely. The opposite effect is seen in the cardiac isozyme, where phosphorylation activates the kinase and inactivates the phosphatase. Skeletal muscle PFK2, interestingly, isn't covalently regulated by phosphorylation.
Different tissues control it differently based on their needs. Very clever.
This differential regulation ensures that each tissue can precisely fine-tune its glycolytic rate according to its specific metabolic needs. What's more, fructose 2-6-bisphosphatase also acts as a potent inhibitor of fructose 1.6-bisphosphatase.
Wait, fructose 1-6-bisphosphatase? That sounds like it does the reverse of PFK1.
It does. That's a key enzyme in gluconeogenesis, the pathway for making new glucose. So fructose 2-6-phosphate activates glycolysis, PFK1, and inhibits gluconeogenesis.
Ah, reciprocal regulation. Prevents breakdown and synthesis happening at the same time.
Exactly. This reciprocal action is brilliant. It prevents a futile cycle where glucose is broken down only to be immediately resynthesized, ensuring maximum metabolic efficiency and preventing energy waste.
And how do hormones tie into this PFK2 and fructose 2.6-bisphosphate system?
This hormonal control is a beautiful example of your body's long-term energy management. In the well-fed state, After a carbohydrate-rich meal, you have decreased levels of glucagon and elevated levels of insulin.
High insulin, low glucagon.
In the liver, this hormonal shift causes PFK2 to become dephosphorylated. Based on what we just said, what does that do to its activity?
Dephosphorylated in the liver, that means the kinase activity is on N, phosphatase is O off F, so it makes more fructose 2.6 bisphosphate.
Perfect. This leads to an increase in hepatic fructose 2.6 bisphosphate and consequently a significantly increased rate of glycolysis via PFK1 activation. So fructose 2.6 bisphosphate acts as a crucial intracellular signal of glucose abundance, telling the cell to burn glucose for energy or convert it to storage.
Okay, so well-fed state, more fructose, 2.6 bisphosphate needs more glycolysis. What about fasting?
Conversely, during fasting, when glucagon levels are high and insulin levels are low,
High glucagon, low insulin.
PFK2 in the liver becomes phosphorylated via glucagon signaling through CAMP and pKa. What does phosphorylation do to liver PFK2?
Phosphorylation in the liver. Kinase, O off F, phosphatase on N. So it breaks down fructose 2-6 bisphosphate.
Exactly. This leads to a decrease in fructose 2-6 bisphosphate, which inhibits glycolysis, less activation of PFK1, and simultaneously relieves the inhibition on gluconeogenesis, since fructose 1-6 bisphosphatase is no longer inhibited.
So fasting less fructose 2-6 bisphosphate equals less glycolysis, more glucose synthesis. Wow.
It's a brilliant system for ensuring your body is either efficiently breaking down glucose for immediate energy when it's plentiful, or making new glucose to maintain stable blood sugar for vital organs when food is scarce.
And just to recap the energy costs so far, we used 1 ATP with hexakinase glucokinase and 1 ATP here with PFK1, so 2 ATP invested.
Correct. 2 ATP consumed during this entire energy investment phase, priming the glucose, now as fructose 1-6 bisphosphate, for the larger energy harvest to come.
Okay, what happens to fructose 1-6 bisphosphate now?
Following PFK1's action, fructose 1-6 bisphosphate cleavage occurs. The enzyme aldolase cleaves the 6-carbon fructose 1-6 bisphosphate into 2 distinct 3-carbon molecules, dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate.
It splits the 6-carbon sugar into 2 3-carbon sugars, DHAP and glyceraldehyde 3-phosphate.
This reaction is readily reversible and, unlike PFK1, is not a regulated step in glycolysis. Interestingly, aldolase B, the hepatic isoform of this enzyme, also cleaves fructose 1-phosphate and plays a specific role in how your liver processes dietary fructose.
So 2 3-carbon molecules now, do they both continue in glycolysis?
Good question. Immediately after cleavage, there's a crucial isomerization of dihydroxyacetone phosphate. Triose phosphate isomerase rapidly interconverts DHAP and glyceraldehyde 3-phosphate.
DHAP turns into glyceraldehyde 3-phosphate. Why?
This is vital because only glyceraldehyde 3-phosphate can proceed further into the glycolytic pathway. By swiftly isomerizing DHAP to glyceraldehyde 3-phosphate, this step ensures that both 3-carbon cleavage products of fructose 1-6 bisphosphate are metabolized by glycolysis.
So everything gets funneled into the glyceraldehyde 3-phosphate form.
Exactly. This means that for every 1 original glucose molecule, you get 2 molecules of glyceraldehyde 3-phosphate moving into the next energy-generating phase.
2 molecules entering the payoff phase.
It's also worth noting that DHAP has another important metabolic fate. It can be used in triacylglycerol synthesis, directly linking glycolysis to fat storage pathways.
So it can be diverted to make fat too. Interesting link. Okay, we've put in 2 ATP molecules in the investment phase. We have 2 molecules of glyceraldehyde 3-phosphate, and now we're finally ready for the payoff, right? The energy generation phase.
Let the harvest begin. The subsequent reactions of glycolysis constitute the energy generation phase. This is where a net of 2 ATP molecules are formed per glucose molecule, not from the electron transport chain, but directly via a process called substrate-level phosphorylation.
Substrate-level phosphorylation, meaning the energy comes directly from the molecule being processed.
Precisely. It's an immediate direct energy transfer from a high-energy intermediate to ADP to make ATP.
Okay, first step of the harvest.
The first pivotal reaction in this energy-generating phase is the oxidation of glyceraldehyde 3-phosphate. Remember, we have 2 of these per glucose.
Right, 2 molecules going through this.
This conversion to 1,3 bisphosphoglycerate, or 1,4,3-BPG, catalyzed by glyceraldehyde 3-phosphate dehydrogenase, is the first oxidation-reduction reaction of glycolysis.
Right.
Shown in figure 8.18.
Oxidation. So electrons are removed. Where do they go?
A key part of this reaction is the formation of NADH. NAD plus accepts the electrons and becomes NADH.
Ah, NADH is formed here. That's energy storage too,
That
right?
is significant energy storage.
Yeah.
But this is absolutely critical because the limited amount of NAD plus in the cell means that the NADH formed must be reoxidized back to NAD plus for glycolysis to continue. The cell can run out of NAD plus otherwise.
So you need to recycle the NAD plus diapate. How does that happen?
Your body has two major mechanisms for doing this. Under anaerobic conditions, like during intense exercise, NADH is reoxidized by the reduction of pyruvate to lactate, a process catalyzed by lactate dehydrogenase. We'll talk more about that later.
Okay, lactate formation recycles NADH anaerobically. What about aerobically?
Under aerobic conditions, when oxygen is plentiful, NADH is reoxidized by the powerful electron transport chain in the mitochondria. However, NADH itself cannot directly cross the inner mitochondrial membrane.
Right, it's stuck in the cytosol where glycolysis happens.
So it requires specific substrate shuttles, like the malate aspartate and glycerol 3-phosphate shuttles, to move those reducing equivalents, those electrons, into the mitochondrial matrix where the electron transport chain is.
Shuttles to get the energy from NADH into the mitochondria.
Okay. What's particularly clever about this specific step, the glyceraldehyde 3-phosphate dehydrogenase reaction, is how it conserves energy. The oxidation of the aldehyde group of glyceraldehyde 3-phosphate to a carboxyl group is directly coupled to the attachment of an inorganic phosphate, pi, from the cytosol to that carboxyl group.
So oxidation and adding a phosphate happen together.
Yes, forming a high-energy bond in the product, 1,4,3-bisphosphoglycerate, 1,4,3-BPG. This high-energy phosphate bond conserves a significant portion of the free energy produced by the oxidation.
Storing the energy from oxidation in that phosphate bond.
Exactly. And the stored energy is then elegantly used to drive ATP synthesis in the very next reaction of glycolysis. It's beautifully coupled.
And this specific reaction is a great place to highlight something grim but biochemically fascinating, the effects of arsenic poisoning.
Yeah, arsenate, the pentavalent form, specifically messes this step up.
We're not talking about just any arsenic, but pentavalent arsenic or arsenate. While trivalent arsenic inhibits other enzymes, arsenate has a more insidious effect directly on glycolysis. It can prevent net ATP and NADH production by glycolysis without actually inhibiting the pathway itself.
How? Here's the molecular trick.
Yeah.
Arsenate looks chemically similar to inorganic phosphate, pi. So it competes with pi as a substrate for glyceraldehyde 3-phosphate dehydrogenase.
It pretends to be phosphate.
Exactly. Instead of forming the high-energy 1,3-BPG, the enzyme mistakenly incorporates arsenate, forming an unstable complex that spontaneously hydrolyzes, breaks down, to 3-phosphoglycerate.
So it makes the next product 3-phosphoglycerate, but skips the high-energy 1,3,3-BPG intermediate.
Precisely. This clever mimicry bypasses the synthesis of 1,3,3-BPG and, critically, the subsequent transfer of its high-energy phosphate to make ATP in the next step.
Whoa, so glycolysis continues, but you lose the ATP payoff from this step.
You got it. It essentially uncouples the oxidation from ATP formation at this stage, depriving the cell of the energy it would normally gain. Arsenate also complicates things further by competing with pi binding to ATP synthase later on, further disrupting the cell's ability to generate energy.
A chilling example of molecular mimicry with devastating physiological effects.
Absolutely. Related to 1,3-BPG, there's a fascinating side reaction, particularly important in red blood cells. The synthesis of 2,3-bisphosphoglycerate, 2,3-BPG, not 1,3-BPG, not 1,3-BPG. Right, the phosphate moves. While only trace amounts are found in most cells, 2-around-3-BPG is present at very high concentrations in red blood cells. Some of the 1,333-BPG produced in glycolysis is converted to 2-ary-3-BPG by the enzyme bisphosphoglycerate mutase.
Why do red blood cells need so much 2-1,3-BPG?
This 2-ary-3-BPG serves a crucial function. It binds to hemoglobin and serves to increase oxygen delivery from hemoglobin to tissues. It helps hemoglobin release oxygen where it's needed.
Ah, it modulates hemoglobin's oxygen affinity.
Exactly. It's then hydrolyzed by a phosphatase back to 3-phosphoglycerate, which can re-enter glycolysis. This highlights how glycolysis in red blood cells is uniquely modified to serve their specialized physiological demands beyond just ATP production.
Okay, back to the main pathway. We make 1,3-BPG, 2 of them. What's next?
Next, we move to the synthesis of 3-phosphoglycerate and the first ATP production in the energy-generating phase.
Finally making some ATP.
When 1-vol-3-BPG is converted to 3-phosphoglycerate, that high-energy phosphate group we just discussed is directly transferred to ADP to synthesize ATP. This reaction is catalyzed by phosphoglycerate kinase, as seen in figure 8.18.
Substrate-level phosphorylation again.
A prime example. The energy for ATP production comes directly from the substrate, 1-3 BPG. And here's the crucial ATP balance point. Since two molecules of 1,503 BPG are formed from each original glucose molecule.
This reaction produces two ATP molecules.
Right, which replaces the two ATP molecules consumed in the initial energy investment phase.
We're finally breaking even on our ATP investment. Net zero ATP so far.
Correct, net zero at this point. After that, there's a relatively simple but necessary phosphate group shift. Phosphoglycerate mutase catalyzes the freely reversible shift of the phosphate group from carbon-3 to carbon-2 of phosphoglycerate.
Just moving the phosphate over. Why?
It's a straightforward rearrangement, merely positioning the molecule correctly for the next energy-generating step, creating another high-energy phosphate bond.
Okay, setting up the next payoff. What happens to 2-phosphoglycerate?
Then we have the 2-phosphoglycerate dehydration. The enzyme enolase dehydrates 2-phosphoglycerate, removing a molecule of water to form phosphenylpyruvate, or PP.
PP. That sounds high energy.
It is. This molecule contains a high-energy enol phosphate, as shown in figure 8.18, making it highly unstable and perfectly primed to donate its phosphate in the final step. This reaction is also reversible, despite the high-energy nature of the product.
And here's a direct clinical connection you mentioned. Fluoride.
Yes, fluoride actually inhibits enolase. This explains why water fluoridation helps reduce lactate production by mouth bacteria, by inhibiting their glycolysis, thereby decreasing dental caries or cavities.
Wow, fluoride works by blocking this specific enzyme in bacterial glycolysis.
It's a direct link from a seemingly small molecular step to everyday health and preventative care.
Okay, we've made the high-energy PPP molecule, two of them. What's the final step?
Finally, we arrive at the grand finale of glycolysis, pyruvate synthesis, and the second ATP production via substrate-level phosphorylation.
The final payoff.
The conversion of PP to pyruvate, catalyzed by pyruvate kinase, or PK, is the third irreversible reaction of glycolysis. The high-energy enol phosphate in PP is leveraged to synthesize ATP directly from ADP.
Another substrate-level phosphorylation. And since there are two PP...
This step generates two more ATP molecules. Since we broke even earlier, this gives us a net gain of 2 ATP for the entire glycolytic pathway per glucose.
Net gain of 2 ATP, plus the 2 NADH we made earlier. Okay. How is pyruvate kinase regulated? It's irreversible, so probably controlled.
It is. Pyruvate kinase activity is itself subject to feed-forward regulation, which is an elegant control mechanism. It's activated by fructose 1.6 bisphosphate.
Fructose 1.6 bisphosphate. That's the product of the PFK1 reaction way back.
Exactly. This ingenious feed-forward mechanism links the two key kinase activities.
Yeah.
If PFK1, the first committed step, is highly active, it produces a lot of fructose 1.6 bisphosphate, which then directly activates PK further downstream.
So PFK1 basically tells PK, hey, lots of stuff is coming your way, get ready.
That's a great way to think about it. It ensures that when the first committed step of glycolysis speeds up, the entire pathway follows suit efficiently. Pyruvate kinase is also allosterically inhibited by high levels of ATP.
Makes sense. High energy shuts it down too.
Another sensible way to slow glycolysis when the cell has abundant energy, preventing unnecessary glucose breakdown.
Now, you mentioned earlier that the liver regulates things differently sometimes. Does that apply to pyruvate kinase too?
Yes. In the liver, pyruvate kinase also undergoes covalent regulation, adding another layer of control. When blood glucose levels are low, such as during fasting, there's an elevated level of the hormone glucagon.
Glucagon again, high during fasting.
Glucagon, through the CAMP pathway we discussed earlier, activates protein kinase A, pKa. This pKa then phosphorylates and, crucially, inactivates the hepatic isozyme of pyruvate kinase as depicted in figure 8.19.
So glucagon causes PK in the liver to be turned alpha-FF via phosphorylation.
Correct. The consequence. With an inactivated PK, phosphenylpyruvate, or PEP, is prevented from continuing down the glycolytic pathway to pyruvate. Instead, it gets shunted into the gluconeogenesis pathway.
To make new glucose.
Right. This mechanism beautifully explains how glucagon both inhibits glycolysis, glucose breakdown, and stimulates gluconeogenesis, glucosynthesis, in the liver, helping your body maintain stable blood sugar levels during periods of fasting. Conversely, when insulin is high and glucagon is low, a phosphatase removes the phosphate, reactivating PK and allowing glycolysis to proceed once more in the liver.
Incredible hormonal coordination switching between pathways.
This brings us to a significant clinical condition, pyruvate kinase deficiency.
Yes, you mentioned this affects red blood cells particularly. Why them?
Mature red blood cells are uniquely affected by this deficiency because they lack mitochondria, meaning they are completely dependent on glycolysis for their entire ATP production. They have no backup energy system.
No mitochondria, no oxidative phosphorylation. Glycolysis is their only option.
Exactly. ATP is absolutely vital for red blood cells to maintain their flexible, biconcave shape, which allows them to squeeze through narrow capillaries and to fuel the ion pumps that regulate their internal environment.
What happens when PK is deficient and they can't make enough ATP?
When glycolysis is reduced due to PK deficiency, there's a significant decrease in ATP production via substrate-level phosphorylation. This leads to alterations in the red blood cell membrane, changes in cell shape, they become less flexible, more fragile, and ultimately their premature destruction by splenic macrophages,
The spleen filters out the abnormal cells.
leading to mild to severe non-serocytic hemolytic anemia, meaning anemia caused by red blood cell breakdown. Severe forms might even require regular blood transfusions. The majority of inherited glycolytic enzyme deficiencies are, in fact, due to PK deficiency.
Why aren't liver cells affected if it's the same gene?
It's fascinating. While liver PK is encoded by the same gene, liver cells are generally unaffected despite having the deficiency because they can synthesize more PK to compensate, and crucially, they can also generate vast amounts of ATP through oxidative phosphorylation in their mitochondria. They have backup systems.
Red blood cells don't have that luxury.
The severity of the anemia depends on the degree of enzyme deficiency. Some residual activity usually remains, and how well red blood cells can compensate by synthesizing increased levels of that 2-MAR3 we talked about earlier, which can sometimes help stabilize things.
And there was a malaria connection.
An intriguing fact: individuals heterozygous for PK deficiency, meaning they have one normal and one mutant copy of the gene show resistance to the most severe forms of malaria, suggesting an evolutionary advantage in certain populations where malaria is prevalent.
Wow. So this step, pyruvate kinase and its potential dysfunctions, offer a powerful insight. It's a critical control point, directly tying your cell's energy demands to its glucose utilization.
And highlighting tissue-specific vulnerabilities.
Its malfunction, as we just discussed, can lead to serious systemic health issues like anemia, highlighting just how finely tuned this seemingly simple pathway needs to be for optimal health and survival. It's a testament to the domino effect of cellular processes. Hashtag epyruvate reduction to lactate, anaerobic glycolysis. Okay, so glycolysis ends with pyruvate, 2 molecules per glucose. We've touched on this, but let's explore one of pyruvate's crucial immediate fates, its reduction to lactate, especially under anaerobic conditions.
Right. This is the endpoint of anaerobic glycolysis. Lactate, formed from pyruvate by the enzyme lactate dehydrogenase, LDH, is the final product of anaerobic glycolysis in eukaryotic cells, as shown in Figure 8.21.
Why is this necessary? What does converting pyruvate to lactate achieve?
Remember the NADH produced by glyceraldehyde 3-phosphate dehydrogenase. Under anaerobic conditions, the cell needs a way to regenerate NAD+ from that NADH so glycolysis can continue. Converting pyruvate to lactate achieves exactly that. LDH uses NADH to reduce pyruvate, producing lactate, and crucially regenerating NAD+.
It solves the NAD+ recycling problem when there's no oxygen for the electron transport chain?
Precisely. This process is absolutely vital when oxygen is scarce, providing a rapid, albeit less efficient, source of energy by allowing glycolysis to keep running.
Where does this anaerobic glycolysis mainly happen?
It primarily occurs in tissues that are poorly vascularized, meaning they don't have a great blood supply and thus receive less oxygen, such as the lens and cornea of your eye or the kidney medulla. It also happens in cells that completely lack mitochondria, such as your red blood cells, which, as we noted, rely solely on glycolysis for their ATP.
And in muscles during hard exercise, right?
Yes, one of the most common and relatable examples of lactate formation is in exercising skeletal muscle. During intense exercise, your muscle's rate of NADH production, both from glycolysis and potentially from other sources, can actually exceed the oxidative capacity of your electron transport chain, even if some oxygen is present.
The demand for ATP outstrips the oxygen supply rate.
Right. This means there's a buildup of NADH and a relatively lower amount of NAD plus RAND plus bed, creating an elevated NADH-NAD plus ratio. This high ratio strongly favors the reduction of pyruvate to lactate by LDH.
And that buildup causes the burn.
As lactate accumulates in your muscle, it causes a drop in intracellular pH. It's produced as lactic acid, which dissociates, which you feel is that burning sensation, and can potentially contribute to muscle fatigue and cramps. Much of this lactate then diffuses out of the muscle cells into your bloodstream.
Is lactate just a waste product then?
Not at all. Lactate isn't just waste, it's also a valuable fuel. Its utilization depends on the relative intracellular concentrations of pyruvate and lactate, as well as the NADH-NAD plus ratio in a given tissue.
So other tissues can use the lactate produced by muscles?
Absolutely. For instance, in tissues like the liver and heart, where the NADH-NAD plus ratio is typically lower than in exercising muscle, lactate taken out from the bloodstream is readily oxidized back to pyruvate by LDH. The reaction is reversible.
What happens to that pyruvate in the liver or heart?
In the liver, that pyruvate has a couple of important options. It can be converted back to glucose through gluconeogenesis. This is called the Cori cycle, recycling lactate back into glucose for other tissues.
Recycling muscle waste back into fuel?
Or it can be converted to acetyl-CoA and then oxidized in the TCA cycle for energy. Heart muscle is particularly interesting because it exclusively oxidizes lactate, converting it back to pyruvate first, then acetyl-CoA, to carbon dioxide and water via the TCA cycle. The heart is very good at using lactate as a fuel, especially during exercise.
The heart loves lactate. Good to know. What about when lactate buildup becomes a problem clinically?
This brings us to a serious clinical condition known as lactic acidosis. This is characterized by dangerously elevated concentrations of lactate in the plasma, leading to a type of metabolic acidosis, lowering blood pH.
What causes that?
It often occurs when there's a collapse of the circulatory system, such as during a myocardial infarction, heart attack, a pulmonary embolism, an uncontrolled hemorrhage, or when an individual is in shock. Basically, any situation causing profound tissue hypoxia.
Lack of oxygen delivery to tissues.
In these dire situations, inadequate oxygen delivery to the tissues leads to severely impaired oxidative phosphorylation and a sharp decrease in ATP synthesis. To survive, cells desperately resort to anaerobic glycolysis for whatever ATP they can get, even if it's meager amounts.
The emergency backup generator kicks in?
Right, but this emergency energy production comes at the cost of producing large amounts of lactic acid, contributing to the acidosis, which can be life-threatening itself.
And this relates to oxygen debt.
Yes. The additional oxygen required to recover from a period of oxygen inadequacy needed to metabolize the accumulated lactate and restore ATP levels via aerobic pathways is often referred to as the oxygen debt. Measuring blood levels of lactic acid is a rapid and crucial early detection method for oxygen debt in patients and is vital for monitoring their recovery and the severity of their condition.
So, what does all this mean for you, the listener? This explains why your muscles burn during a tough workout when they're starved for oxygen. More profoundly, it illustrates how your body has a critical survival mechanism to generate emergency energy when oxygen is scarce, even if it's a less efficient pathway.
It's a remarkable adaptation for acute survival, even if it comes with potential downsides like acidosis in extreme situations. #F energy yield from glycolysis.
Let's quickly recap the energy yield then. We said net 2 ATP, right?
Yes, but it's important to put the energy yield of glycolysis into perspective. While glycolysis does produce some immediate ATP via substrate-level phosphorylation, the end products pyruvate or lactate still hold the vast majority of the energy originally contained within the glucose molecule.
Glycolysis just scratches the surface of glucose's energy potential.
Pretty much. For a complete and maximal release of that energy, the full oxidation through the TCA cycle and subsequent oxidative phosphorylation are required.
So let's break down the actual ATP yield again to clarify the numbers, comparing anaerobic and aerobic glycolysis.
Okay. In anaerobic glycolysis, where glucose is converted to 2 molecules of lactate, there's a net gain of 2 ATP molecules for each glucose molecule processed, as summarized in figure 8.22.
Net 2 ATP. What about NADH?
Crucially, in anaerobic glycolysis, there is no net production or consumption of NADH. Remember, the 2 NADH produced by glyceraldehyde 3-phosphate dehydrogenase are immediately consumed when the 2 pyruvates are reduced to 2 lactates by LDH, regenerating the 2 NAD plus needed to keep glycolysis going without oxygen.
Okay, so anaerobic equals 2 ATP net, 0 NADH net. What about aerobic?
In aerobic glycolysis ending with 2 pyruvates, the net gain of 2 ATP molecules per glucose is the same as in anaerobic glycolysis from the direct substrate-level phosphorylations at phosphoglycerate kinase and pyruvate kinase steps.
Still net 2 ATP directly from glycolysis itself, but what about the NADH?
However, in this pathway, 2 molecules of NADH are also produced per molecule of glucose at the glyceraldehyde 3-phosphate dehydrogenase step, and these are not immediately consumed since pyruvate isn't reduced to lactate.
So aerobic glycolysis equals 2 ATP net plus 2 NADH net.
Correct. For ongoing aerobic glycolysis, most of this NADH needs to be reoxidized by the electron transport chain in the mitochondria, using oxygen as the final electron acceptor. And significantly, each NADH molecule whose electrons enter the electron transport chain via those substrate shuttles we discussed,
The meliaspartate or glycerol 3-phosphate shuttles.
ultimately produces about 2.5 to 3 ATP via oxidative phosphorylation, depending on the shuttle used.
Whoa. So those two NADH are worth another 5 or 6 ATP later on.
Exactly. This is where the bulk of the energy from glucose comes from if oxygen is available, making aerobic respiration vastly more energy efficient for sustained power compared to anaerobic glycolysis alone.
So this clarifies a fundamental energy efficiency principle for your body. The significant difference between glycolysis operating alone versus when it's coupled with oxidative phosphorylation.
Huge difference in ATP yield.
It profoundly shows why oxygen is so vital for sustained, high-yield energy production in your body. It's the difference between a quick burst of energy, anaerobic, and a long, efficient, powerful burn, aerobic, that can fuel complex activities for extended periods.
Oxygen unlocks the vast majority of glucose's energy.
We've talked extensively about pyruvate being reduced to lactate, especially during intense exercise or oxygen deprivation, but you said pyruvate is at a crossroads. What else can happen to it?
Pyruvate is far from a dead-end product. In fact, it's a remarkably versatile molecule that sits at a crucial crossroads in metabolism, with several important fates beyond just lactate.
A central hub.
It truly demonstrates how different metabolic pathways intertwine and adapt to the cell's immediate needs for energy, storage, or synthesis.
Yeah.
Hashtag A, oxidative decarboxylation to acetyl-CoA.
Okay, what's a major fate under aerobic conditions?
One of pyruvate's most significant fates, particularly under aerobic conditions when energy is needed, is its oxidative decarboxylation to acetyl-CoA.
To acetyl-CoA, the fuel for the Krebs cycle.
Exactly. This irreversible conversion is catalyzed by the massive and highly regulated pyruvate dehydrogenase complex, or PDHC, found inside the mitochondria, as shown in Figure 8.24.
PDHC, a big enzyme complex.
This pathway is particularly important in tissues with a high oxidative capacity, such as cardiac muscle, which constantly needs a lot of energy to keep your heart pumping. Acetyl-CoA, the product of this reaction, is a major fuel that enters the tricarboxylic acid cycle for complete oxidation to CO2, generating lots of NADH and FADH2 for oxidative phosphorylation.
Then acetyl-CoA can be used for other things too, right?
Yes, when energy is plentiful, acetyl-CoA also serves as the fundamental carbon source for the synthesis of fatty acids. So PDHC links carbohydrate breakdown to both energy production and fat synthesis. Hashtag B carboxylation to oxaloacetate.
What's another important fate for pyruvate?
Another key fate allowing it to contribute to other metabolic pathways is its carboxylation to oxaloacetate.
Adding a carbon dioxide group.
Yes. This reaction is catalyzed by pyruvate carboxylase, which is a biotin-dependent and irreversible enzyme, also shown in Figure 8.24. This primarily occurs in the mitochondria as well.
Pyruvate carboxylase. Why is making oxaloacetate important?
This reaction is incredibly important because it serves two main purposes. First, it's crucial for replenishing the intermediates of the TCA cycle. Oxaloacetate is itself a key component of the TCA cycle. Sometimes intermediates get pulled out of the cycle for other biosynthetic pathways, so this reaction helps top it up. It's called an anaplerotic reaction.
Anaplerotic, filling up the cycle.
Right. And perhaps even more importantly, oxaloacetate provides a direct substrate for gluconeogenesis, the pathway responsible for synthesizing new glucose from non-carbohydrate precursors when blood sugar levels are low. Pyruvate carboxylase is a key step in converting pyruvate back towards glucose.
So this pathway is essential for making new glucose.
Absolutely essential, especially in the liver and kidney. Hashtag a reduction to ethanol microorganisms.
Okay. Acetyl-CoA, oxaloacetate. Anything else? You mentioned ethanol earlier.
It's also worth a brief mention that in some organisms, specifically yeast and certain other microorganisms, pyruvate can be reduced to ethanol. This is alcoholic fermentation.
Right, how yeast makes beer and wine.
This process involves two steps. First, the decarboxylation of pyruvate, removing CO2, to acetaldehyde by a thiamine-requiring enzyme called pyruvate decarboxylase, followed by the reduction of acetaldehyde to ethanol using NADH, which regenerates NAD+ for their anaerobic glycolysis.
So it serves the same NAD plus regeneration purpose as lactate production in us.
Exactly. However, it's important to clarify that this fascinating pathway does not occur in humans. We lack pyruvate decarboxylase.
Good to know. We make lactate. not ethanol.
Correct.
So this broader perspective on pyruvate isn't just a list of facts. It's a powerful insight into your body's metabolic flexibility. Pyruvate isn't just a simple end product of glycolysis.
It's a major branch point.
Depending on the cell's energy state, oxygen availability, and overall metabolic needs, pyruvate can be channeled into vastly different directions. For immediate energy via acetyl-CoA and the TCA cycle, for replenishing metabolic cycles or making new glucose via oxaloacetate, or for anaerobic energy via lactate.
This adaptability is absolutely key to your body's survival and its remarkably efficient dynamic functioning.
We've spent a lot of time on the rapid, moment-to-moment regulation of glycolysis, allosteric effects, phosphorylation, things happening over minutes or hours. But you mentioned another layer, long-term control.
That's right. Beyond the immediate fine-tuning, there are long-term hormonal effects that influence the number of key enzyme molecules actually present in your cells.
Changing the amount of the enzymes themselves, not just their activity.
Exactly. This is a much slower, yet profoundly impactful form of regulation, fundamentally altering the metabolic machinery itself over longer timescales.
How does that work?
These long-term hormonal effects often involve changes in gene transcription, turning the genes that code for these enzymes up or down. This can lead to 10 to 20-fold increases in enzyme synthesis over periods ranging from hours to days.
Wow, huge changes in enzyme levels. What triggers this?
Hormones and dietary state are major triggers. For example, the regular consumption of carbohydrate-rich meals or the administration of insulin causes a significant increase in the amount of glucokinase, PFK1, and pyruvate kinase in the liver, as illustrated in Figure 8.23.
The three key regulated enzymes of glycolysis we discussed.
Precisely. This increase in the quantity of these enzymes dramatically favors the conversion of glucose to pyruvate, which is characteristic of the absorptive or well-fed state, ensuring your body efficiently processes and stores incoming glucose when it's abundant.
So a high-carb diet or high insulin literally tells the liver build more glycolysis machinery.
Yes. This process is mediated by specific transcription factors, proteins that bind to DNA and control gene expression like steroid regulatory element binding protein 1C, SRBP1C, and carbohydrate response element binding protein, CHREBP, which act as master switches for these genes in response to insulin and glucose signals.
What about the opposite situation, fasting or low insulin?
Conversely, if you're in a state of prolonged fasting, or if you have uncontrolled diabetes, where plasma glucagon levels are high and insulin levels are low, the gene expression of these very same three enzymes, glucokinase, PFK1, and pyruvate kinase, is decreased.
Glucagon tells the liver to make less glycolysis machinery.
Correct. This fundamentally reduces the overall capacity for glycolysis in the liver, effectively shifting the liver's metabolism towards glucose production, gluconeogenesis, rather than glucose breakdown. It's a remarkable long-term adaptation to conserve glucose when supplies are scarce or perceived to be scarce, as in diabetes.
So what's the profound implication of this long-term orchestration for you? This sounds really important for adaptation.
It absolutely is. This explains how your diet and your hormones don't just fine-tune your metabolism from moment to moment, but they fundamentally alter the very machinery of your cells over days and weeks.
It's like changing the factory's equipment based on long-term supply and demand.
That's a great way to put it. They're adapting your body for long-term energy management, determining whether your body is primarily set up to efficiently break down glucose and store energy when it's abundant, or to conserve glucose, release stored energy, and synthesize new glucose when supplies are scarce.
It's an incredible testament to the body's remarkable adaptive capacity to ensure its survival under varying conditions over the long haul.
Deep, fundamental adaptation through gene expression. Hashtag outro.
Wow. We've certainly embarked on an incredible deep dive today. From painting the broad landscape of metabolism as an interconnected, purposeful network,
All those pathways linking up.
to tracing the intricate journey of glucose through the energy investment and generation phases of glycolysis.
Spending ATP to make ATP and NADH.
We've unpacked its different fates, lactate, acetyl-CoA, oxaloacetate, and explored the incredibly sophisticated short-term and long-term regulatory mechanisms that keep it all in exquisite dynamic balance.
Allosteric control, phosphorylation, hormones, even gene expression. Multiple layers.
Indeed. We've seen how individual enzymatic reactions rarely act alone, forming purposeful networks and cycles. We distinguish between catabolism, the breaking down for energy, and anabolism, the building up with energy.
The two sides of the metabolic coin.
And then we journeyed through the meticulous layers of regulation, from immediate intracellular signals like ATP and AMP levels to the complex intercellular communication systems involving hormones like insulin and glucagon, G proteins, and crucial second messengers like HAMPP controlling enzymes like PFK2 and PK.
It's an intricate dance of signals and responses.
Understanding these molecular details provides profound insight into conditions like diabetes, why you experience those sudden energy swings, and even the subtle effects of common substances like caffeine-blocking CAMP-MP breakdown or fluoride-inhibiting analase.
It connects the molecules to real-life physiology and health.
This deep dive should leave you not just informed, but truly well-informed, with a much deeper appreciation for the continuous, orchestrated cellular dance happening within you every single second.
Hopefully feeling like you've got that expert-level grasp we aimed for.
Absolutely. It's truly mind-boggling, isn't it? Given the incredible precision and multiple overlapping layers of regulation we've discussed today, from enzymes like PFK1 acting as master switches, to the subtle differences in glucose transporters dictating cellular access, to the dramatic impact of even small molecular changes like those seen in pyruvate kinase deficiency or arsenic poisoning.
The detail is just astounding.
What does it tell you about the body's remarkable ability to adapt? And perhaps even more importantly, what happens when even one small part of that intricate network goes awry, whether from inherited disease or external factors? How might this deeper understanding of such intricate biochemical processes impact our approach to health, wellness, and even how we view the fuel we put into our bodies?
Really makes you think about the foundation of health at the molecular level.
Something to ponder until our next deep dive.